It is my pleasure to introduce our seminar speaker today, Dr. Warren Sconiers. I met Warren through those all-important networking events at a national entomology meeting. And so that really does pay off, being social and going out. But what I wanted to do some internet trolling to have a good introduction for him, and I honestly didn't find a lot of dirt. The best page is from your current university. And so you have one of those heartwarming, touching stories about how you stumbled into entomology because you loved it ever since you were a kid. So for someone with an innate love for bugs, my first question is, how did you end up at, oh, now it's not working, at UC Irvine, where they're the anteaters? So someone who loves insects right away chose a school that whose mascot is eating ants. But so Warren's originally from California, did his undergrad at UC Irvine. I think he'll tell you more about his experience. But then you moved on to Texas A&M, and that Aggie sure looks like he wouldn't be very kind to any insect that he saw crawling across the floor either. And now you're at the University of the Ozarks, which are the eagles. I don't know, maybe they're a good host for some parasitic insects. So maybe your fondness is changing in the reflection of your mascots. But really, I really admire Warren and the work that he's doing. He has this quote that I pulled off the website when he's talking about teaching and his job, and that interdisciplinary learning is key to critical thinking and global citizenry. And that's what really hits me, like this approach that, you know, we're entomologists, but we have a larger place in terms of our education, our role as educators. And while Warren doesn't have an extension component at this university, you know, he's really engaging with the community on different levels, and sort of those are the things that we really have in common. And so I feel like I roped him into giving seminar because he and I have been working together this semester to host a symposium at the annual meeting. So he and I are both very interested in urban agriculture. And so we are hosting a symposium called Entomology and Urban Agriculture, Growing Food for All. I think he's going to talk today about maybe a lot of actually what you're going to present in the meeting, but I encourage all of you to come attend this, and I will let Warren take it from here. So thank you so much for being here today, Warren, and we look forward to your presentation, and I hope I stopped sharing, so you should be—
You did, yes.
All right.
All right, can everyone see my screen? Yeah, all right. Good morning, everyone. Thank you for attending the seminar. So Laura touched on a few things about my introduction, a little bit about me. Some things I did not expect for her to touch on, but that's good. So today, in urban insect ecology, understanding pollinator immunity and IPM in human environments. And so I'll be talking about my work that I did at NC State University as a postdoc, and also my work that I've been doing at University of the Ozarks. All right, so let's get started. All right, so the outline for the talk for today. So I'll introduce myself, where I came from and what I've been up to. And then I'll look at or discuss immune response in social insects, some work we did at the NC State University, looking at urban pollinators and their response to some temperatures, but also looking at immune response in social lineages, using more of Raleigh, North Carolina as a backdrop for our collection of those insects. So mostly looking at the social lineages and how they have an impact on immunity. After which, I'll be discussing my work at University of the Ozarks, plant architectural complexity and IPM in a community garden. So looking at our work in community gardens, how to maximize yield with crops using plant architectural complexity in order to create more pest control and recruitment of pest consumers. And then finally, just a bonus that I didn't discuss in an abstract is some work I've been doing with undergraduates this semester to get more student research in the classroom and get students outside as much as possible during this pandemic. All right, so first I'll spend some time, a few minutes introducing myself. aside from Laura's lovely introduction. And so I started at University of California, Irvine, ecology and evolutionary biology. And so I worked with Catherine Studing for research. So this was in invasive plant ecology. So looking at how environmental factors can influence how plants are recruited. So how invasive plants come into ecosystems, how disturbance such as fire, how blocking off rain, ambient rainfall can interrupt the recruitment of native plants and then allow for native plants to be able to establish in those systems. Also, we did some work with the University of Colorado Boulder during some summer research looking at how plant phenology and alteration of plant ecology occurs in alpine ecosystems. Then I worked at, or got my PhD at Texas A&M University, working with Mickey Eubanks with drought and plant ecology. So looking at how drought stress affects cotton plants and how those plants are changing in their physiology, their nutritional aspects, and how those changes in turn influence a community ecology with insects. So how do insects respond to that? So are there more insects there? Are there more herbivores there? Are there less, and why? And so looking at the nutritional mechanisms that may have influenced those relationships between drought stress plants and insects. Afterwards, I went to North Carolina State University for a postdoc, working with agriculture and life sciences, assistant dean in particular, and also being hosted in Steve Frank's lab, but working with Lisa Goulian-Jones primarily as, not only did I do research during my postdoc, but I also taught graduate level courses. I did a lot of university service, so started some student organizations, and also worked with the community to get them more involved in science and research. So it was more of the postdoc was research, but also included about half of it was with university service and reaching out to the greater community to incorporate more citizen research and citizen science involved with the university. Okay, so that was essentially where I came from and my background. So now I'm at the University of Ozarks in Clarksville, Arkansas. We're a Presbyterian liberal arts and private university, a four-year university. We have about a whopping 900 students, and that's a record high for us. So across the entire university, we have 900 students. I am one of 4 biology faculty. Just when I first started, I was one of 3. So we actually expanded and now we have 4 biology faculty. Let's check real quick. Okay. Just to make sure we have no microphone problems, I'm going to switch to having no mic, and hopefully that fixes the problem. Okay, Laura, just let me know if this continues to be an issue. So before COVID, we had about 900 students, and that was sort of the best or the highest record we've had for student recruitment on campus. We have 35% international students, so we have a lot of partnerships and associations with South America, the Bahamas and Haiti, and also Europe to recruit more students. And then we're primarily teaching institutions. So 80% teaching is my position, 20% research and everything else. So we teach labs, we advise students, we have a 16-to-1 ratio on campus, and we have small internal research grants. And so that's our university. I'll get into more about about University of the Ozarks because once I get to that section of the project looking just at that work with them. All right, so that was the introduction. Now I'm going to switch gears and look at your immune response using urban social insects in Raleigh, North Carolina. And so looking at social immunity, so social insects in particular, bear the risk of increased infection and disease. So working in close quarters with each other, of course, lots of crowding, it's sort of a great opportunity for pathogens and disease to spread through any sort of population, whether you're humans, you're bacteria, or mammals, insects, etc. Having a close, tight-knit community is just a great opportunity for pathogens and disease to spread through a system. There's inherent relatedness as well, in particular for social insects, in that once a pathogen gets into or able to infect a few individuals, it gets spread like wildfire throughout an entire community because of that inherent relatedness that they all share. And also through feeding and crowding. So of course, large population densities, but also you'll have individuals who will feed each other. Trophallaxis, you'll have sharing of frass for some individuals, like social roaches other individuals. And so you can have that sort of inherent spread of pathogens as well. However, some of the immune functions they have or ways to protect themselves aren't entirely known. And so there's a few hypotheses I'll talk about that sort of try to explore some of the mechanisms behind social immunity. So aside from that, so looking at individual costs, because of you're in such high densities in such a profound way to spread pathogens, metabolic costs for individual immunity can be very high. And so producing enough compounds to weather the storm, if you will, against other kinds or against infection from your nestmates, that can be very difficult to have, and that can cost a lot of energy resources. You also risk the overreaction to your own immune response. Of course, we have allergies, so having such a thing with having large densities of nestmates can be an issue as well. And so selection may actually favor behavioral immunity, so actual learned behaviors that you do rather than metabolic immunity. So especially as colony size increases, you can have these in particular. And so here's a diagram looking at how a pathogen can actually spread throughout a system. So once you have horizontal transmission or direct, or a member brings into the colony an infection, it can spread through just prophylaxis and feeding in contact with other individuals, as it therefore spreads to a mother colony, through vertical transmission, or through a daughter colony, in which you have these interactions further spreading the pathogen throughout the entire colony. So even neighboring colonies, say if foraging individuals leave and go to a flower, you have some social insects, bumblebees and honeybees, for instance, exchanging these pathogens as they share a similar flower. Whereas with ants will typically guard nest entry or look at different foraging trails or guard their territory in order to prevent this. Whereas social bees and honeybees will use a guarding system in order to protect themselves from external factors. And so with social behavior and structure, you can have protection of foraging trails and resources. You can have guarding of hive entrances as more of a social or behavioral trait that can protect them from infection. And also individuals abandoning the infected. So being able to smell and detect your nestmates being infected and then being able to rid them out of the colony before it spreads to other individuals. So these are all behavioral traits that have been selected for in order to create more protection for the entire colony. So looking at metabolic defense, you can have ingestion of antimicrobial substances from the environment so gathering external protective factors to protect yourself, but also you can produce them yourself. So ants will produce antimicrobial and also get them from their farming. If you're leafcutter ants, for instance, to create a way to protect yourself from your potential pathogen infectious agents. And so another example of this is looking at dampwood termites and some infectious agents, entomopathogenic fungi. And so in this example here, percent survival of dampwood termites, I'm looking at exposure of these termites to this fungus. And so looking at this infection with dampwood termites, and so what they found was that individuals who are, so for instance, this close circles are control without any sort of infection and with just termites, and then looking at a middle dose or low dose of this fungus and having compared to a high dose, and then this is percent survival. So individuals with that were infected by this fungus were actually doing a lot better while they were in social groups. So individuals who were alone with control, alone with a middle dose, and then alone with a high dose have a much more decreased percent of survival while they are alone. Whereas when they were together with their colony, living with their colony mates, having that protection and having those behaviors and grooming, allo-grooming, for instance, taking care of each other, you have a much better survival rate with those individuals. So just with this example alone, you can see that behavioral traits or behavioral immunity provides a lot of protection for individuals compared to being solitary. So this supports the idea that solitary protection, metabolic costs are too high for individuals, and that perhaps selection has moved towards having a social protection or behavioral protection with individuals. And so the social group hypothesis suggests that social insects are selected for greater immune responses compared to solitary insects. And this is despite behavioral immunity. So there is behavioral immunity that takes place, but this hypothesis argues for having more of a greater immune response as an individual compared to your group dynamic. And then the relaxed selection hypothesis suggests the other way, where behavioral immunity reduces disease risk and it reduces cost of immune function. And so instead of relying upon individual immunity, you can use your group dynamic to protect you. And so you don't have to have such metabolic costs invested to protect yourself. And so this is looking at physiological or individual immunity versus behavioral investment in immunity in social insects. So this is exploring two of these hypotheses that look at this idea in order to see, well, is it more of an individual response or is it more of a social response or a behavioral response? And so one way to look at this is, or one way we looked at this is using encapsulation response. So what this is that you have hemocytes that will attach to a foreign object. And this foreign object can be a parasite, a large parasite, or it could be a series of parasitic eggs or offspring getting ready to hatch. And so the hemocytes and also melanin will surround this object and essentially suffocate it, quarantine it off in a particular part of the insect. And then some insects will actually able to expel it out of their tissue, whereas some will simply expel it out through excretion or just isolate it until it becomes simply inert and it is excreted in a molt, for instance, or sloughed often in the exuvium of a molt. And so you can do this by simply adding a foreign object. So this has been done with plastic beads. We use a nylon fiber or nylon filament for this action. And so hemocytes bind to large objects. Parasites are parasitic eggs. It's a cellular test of insect immunity. And so higher encapsulation rates would suggest in social insects that support these social group hypothesis. So if you have more encapsulation in social insects, it would actually support the SGH. But higher encapsulation rates in solitary insects would support the RSH, or the relaxed selection hypothesis. And so phylogenetic generalized linear mixed models were used to determine cellular immune response, to see if behavior was a factor. So we use these models to determine the effect on cellular immunity with behavior, whether it was eusocial or not. Colony size, so was it a small colony versus a large colony? So we combined that with eusociality because there was a large gradient or sort of a large gradient of sociality that we saw across some of the lineages we collected. And so we were looking at using colony size as a complementing factor in our models. We also looked at incubation temperature. So with urban temperatures, we're looking at higher versus lower temperatures to see if that also made a difference with social insects in urban environments. And we also looked at body size. So of course we have larger individuals versus smaller individuals. So we want to standardize for bodies. So we looked at these four variables and how they were able to determine or influence cellular immune response using encapsulation as that response variable. And so some of the methods, so I worked with Margarita Lopez Ribe, who was another postdoc when I was at NC State. And we looked at 11 different insect lineages collected from Raleigh, North Carolina. So downtown, some of the outskirt areas, so urban, suburban, and rural areas, some of these insects that we were collecting. And for the encapsulation protocols, we anesthetized them on ice. And so we would collect them outside, bring them into a cooler, and then we put them in either a freezer or on ice for 20 minutes. For some of the larger insects, we had to go a little bit longer. Some of the smaller insects, sometimes a little bit of less time. That would make them non-responsive. They would sit there. They wouldn't try to fly away, for instance. So it was a simple way to keep them still. And so between the tergites on the abdomen, we're able to insert a 3 millimeter nylon filament, which was the probe. We covered it in a lipopolysaccharide, so to further induce this immune response. So the filament itself would induce a response, but it's been shown that lipopolysaccharide is typical of some coverings for parasitic insects, and so that further induces the immune response. There's a 4-hour incubation period at either 25 or 35 degrees Celsius, 35 degrees initiating or simulating a higher temperature during summer. And the probes were photographed with a microscope camera with a Nikon SMZ 800 camera. And so we're able to take pictures of these probes, and then we use image J to quantify the degree of grayness. So as the insects would coat their probes, you'd have a increasing degree of darkness, and then we can use that, we could quantify that in order to have a quantification of that response color for encapsulation. And so here are some photos of what they look like. And so this is the actual picture. And so the probe is barely present, as you can see in the camera. And then here's the darkened area. And so we would inverse it, invert the colors to make it more able to quantify. And so in a different photo, we have the probe. And so here's a strong immune response. So this whole region here would be quantified as far as the, in this case, the brightness of that color, which would indicate the encapsulation response. And so our response, so that was our response to encapsulation, but looking at the variables that would influence encapsulation. So we looked at eusociality and non-eusocial based on colony size. So just having yes or no or binary component was not enough. And so we want to couple that with colony size, where a larger colony would be increasingly more eusocial and a smaller colony size would be less and less or closer to non-eusocial. And so we also use body size as a continuous variable as well, because the probe size would vary, or not vary, but the amount of hemocytes possible would vary based on the quantity of bloods and because of the body size. And so we wanted to make sure that was standardized. So we use the maximum likelihood of phylogeny. We created that based on 7 genes because of the non-independent evolutionary history of some of these lineages collected. So we wanted to make sure that we had another factor to help us separate them within the phylogeny besides just what we had named them taxonomically. We use a deviance information criterion for Bayesian models for a variability and immune function. So we wanted to look at these models and sort of within those models through the Bayesian models, which ones were actually able to predict variability the best. And then we use phylogenetic generalized linear models to quantify the encapsulation rate. And so for the results, these are different types of lineages we collected. So we had your honeybees, we had several kinds of bumblebees. Then we also had several solitary bees, and we had ants as well that were also that are known for, of course, pollinating, but they were also used in this context for just another social lineage. And then just to make sure it wasn't Hymenoptera, that was the biggest influence. And so we want to also have the termites, and we also had just another outlier or another different individual German roaches. And so we wanted to look at these individuals to see most of these collected, or all of them collected either in urban, suburban, or outskirt areas. And then we had different degrees of sociality among these groups. And so what we found was that, so this yellow is for eusocial and the green is for non-eusocial. And so we found a similar encapsulation between eusocial and non-eusocial lineages. And so encapsulation response is a degree of strength increasing from 0 to pass through 180. So you can see that they overlap quite a bit. So we weren't able to find a significant difference between these two types of general sociality groups. But we did see that colony size, eusociality, and body size create variability in the encapsulation response. And so with the other groups, we saw that honeybees, for instance, you have more of a lower encapsulation response, higher in a less social insect. Solitary, a little bit higher, and then also it ranged quite a bit with a little bit of a little bit of difference there as you look at the green being non-social and the yellow being social. And so there's some individuals who are varying quite a bit. And so it wasn't a very strong difference, but we were able to see some significant details. So we saw that body size was influential, but no interactions with other variables. And so we were able to see, though, that body size made a difference. So looking at the size of individuals changed encapsulation response, of course, but we weren't able to see that with temperature, or we weren't able to see that with sociality. So that was a separate influential factor for looking at immune response. So here are some other individuals. And so some more individuals on the yellow, who are yellow on the lower side, and some individuals who are non-social, a little bit higher than that. So there are some differences overall that we saw between eusocial insects and non-eusocial. So another model detected lower encapsulation with larger colony size. And so looking at the colony size as a factor or a factor of eusociality, that we're able to see larger colony size being a stronger influence rather than eusociality. So categorizing them, I guess, on increasing colony size led to actually a lower encapsulation response. And so what we saw was that encapsulation decreases with increasing colony size in support of the relaxed selection hypothesis. Larger colonies rely on behavioral immunity, whereas smaller colonies or solitary insects rely on more individual immunity. We've seen in several other studies that fewer genes for metabolic immunity in social insects. And so they're, even when they split off through their ancestral history, you'll see that there are genes looking at or showing fewer individual immunity compared to having more behavioral traits. So studies also suggest that stronger antimicrobials are used to complement behaviors in larger colonies. So some individuals actually have one aspect of metabolic immunity, whereas most of their selection has been focusing on having behavioral immunity in response. So more to me than our metrics of eusociality can capture. So it isn't really about, so of course we categorize them as eusocial or non-eusocial, but it looks like more colony size is more important in predicting encapsulation response and therefore behavioral immunity traits. So variation that we saw across our studies was that species-specific created a lot of species-specific situations with encapsulation response was a strong factor. And so creating that created a lot of variation in our data, but also temperature was not as strong as we had hoped. So we thought that temperature in urban systems would be a strong influence compared to non-urban systems, urban versus rural systems. But we saw that the temperature itself was not as important, but that may have been complex with nutritional issues that we know occur in urban systems, such as a lack of diversity of flowers and nectar and pollen to use. But also a lack of diversity just in plants and patchy habitats. So that can also complex with temperature as being more of a correlate with temperature in those issues compared to having just an individual experiencing temperature alone. So future studies would need to look at sort of separating those factors out and seeing if we can see more behavioral issues alone versus the metabolic response to the encapsulation being more of a factor. And so overall, we saw that encapsulation response, this immune factor, was more important with colony size as a way to support the relaxed selection hypothesis. So more behavioral immunity is more important compared to independent or individual immunity in social insects. All right, so that is the end of that first section. So I'll take a brief pause if anyone has any questions right now that they may want to raise. And then if not, then we will continue.
I have a quick one, Warren. Can you just comment on the range in size of the colonies that you were looking at? So it went all the way from solitary to honeybees, which is huge, but what did it look like in the middle?
So yeah, some couple hundred individuals for colonies. Some of the bumblebee colonies were, of course, a lot smaller than some of the larger, more like the termites or the honeybees. And so that was sort of the range that we saw with those individuals. So it wasn't, so of course we had more on the larger side overall, and then a big gap between them and the smaller size.
Thank you.
All right, I'll continue on. So I'll continue with some of the work I did at University of the Ozarks, looking at plant architectural complexity and IPM in the community garden. And of course, questions, all questions at the end for all projects, of course. All right, so looking at this project, so Arkansas is actually second in the nation for food insecurity. So what that means is that, it looks like one popped up for the Q&A, just a second. So do I have evidence of low immunity bees collecting pollen from particularly toxic plants to compensate for the lack of immune response? That's a good question. So I'll answer this live. Let's see. Oh yeah, so a lot of social insects will, or also just insects in general, will actually change their behavior to eat different. So caterpillars will eat different plants in order to get different kinds of compounds to help them fight against parasites, ingesting toxins that they don't typically eat in order to have a way to intoxicate the parasites inside, trying to reproduce inside. Response. However, with social insects, I have not seen too many studies that have, for instance, looking at pollen, but I know that plants will have increased toxic compounds in nectar. So even just as a response to drought or response to less than optimal conditions, plants will actually put more defensive compounds within their nectar, and for several reasons. One is to get rid of or deter non-essential pollinators or pollinators that don't specialize in them, and actually to help the specializing pollinators. But also just as a general response to stress, plants will do that. So there may be a possibility between that link with those pollinators using those defensive compounds to help themselves out against suboptimal conditions. But there aren't many studies I've seen personally that would look at that to see if they're purposefully looking at that type of those kind of plants in order to boost or support their lack of immunity. Okay, there's another question here. Let's see. So if I interpret this correctly, you would focus on field-caught samples, foragers, for each species in part 1. Do you suspect that including different social roles versus queens, et cetera, would change your results when comparing social versus solitary immunity? Yes, that was definitely the case. Some of the literature suggests that not only is that important, but there are actually some behavioral mechanisms that will predict. So there are some studies. So there's a review that was suggesting that looking at the mechanisms in which it takes to take those compounds and eventually get them to the queen is a little bit longer, and there's actually more behavioral mechanisms in the way of that. So before the pathogen even gets into the colony, you'll have some guards that will detect that and get rid of those individuals, like I mentioned before. But you'll have workers inside that are sort of garbage handlers, where they will find workers that have been infected before reaching more important or critical members of the colony and detect those and get rid of them prior to that. And you'll have some cases where you'll have more external workers and operators who are passing around those infectious agents, and those can be detected before they circle in onto the important members, including the queen and some of the secondary reproductives for termites. And so those can be some aspects of behavioral. There's more behavioral protections, you can say, before it gets to more important members of the colony. So definitely would have different results, especially because foragers, those are the older individuals of colonies, especially for honeybees. And so those have a more aged individual immune system anyway. And so younger individuals may have more resistance, may have a stronger immune response, encapsulation response, but also that being even stronger as younger individuals typically stay in the colony for longer compared to older individuals. Okay, so I will continue, and then there's more questions about the first section, and I can answer them there. All right, so Arkansas is actually second for food insecurity, and so only with Mississippi being the first in the nation. So 1 in 4 children and 1 in 6 women live in poverty, and so one of these issues is trying to find a way to support families in Arkansas by providing ways to increase the output of health and fresh and healthy food, but also make it more available and cheaper for lots of families. Lots of families will have access to a lot of cheap, subsidized junk food that is typical for impoverished areas. And so what we want to do is find a way to educate and find a way to provide more ways to increase the exposure of people to healthier types of food or healthier foods. So Johnson County in particular, so here's Arkansas. I'll just give you an orientation, Louisiana to the south, You have Texas and Oklahoma in this direction to the west, and you have Missouri to the north and Tennessee to the east. And you will have, so within Arkansas, we're towards the north-south, on the northwest region of Arkansas. And Johnson County is described by the USDA as a food desert in which households have limited access to fresh, healthy, and affordable food. So community gardens across the state, and in particular our area, are a way for people to have independent food production, income, and education. So we use those gardens as well to have students out there volunteering, getting research credit as well. And so 3 community gardens in Clarksville alone, which is a very small city of about 10,000 people, provide food for domestic and international community members. So we have people who come in who work for the Tyson chicken plant as workers, and they bring community gardens as well to establish. And so there's several different community gardens within Arkansas or within Clarksville alone that help with this issue. And so at University of Ozarks, we have our community garden is called the Food for Thought Garden. Students will do research there for course credit, for independent research, for internships, and for community service. We'll have the AmeriCorps come out there every once in a while, Peace Corps, to also increase awareness of community garden and food, lack of food issues. And so, for instance, community gardens in Johnson County provide education and food. Pesticide applications, though, are commonly used to increase yields. So we want to have more organic, sustainable farming. And so we want to focus more on that and educate more with that when it comes to these types of food production. And so how can the community move to produce more pesticide-free food? And so we wanted to introduce IPM, or integrated pest management with natural enemies, arthropods, looking at arthropod predators, competitors to outcompete pest insects on our crops, community gardens, in order to have better control or non-pesticide-based control. And so we've been discussing these issues through coursework, through community services, about spiders, lacewings, ladybugs that can increase the pest resistance of crops. And then this will promote sustainable agriculture and organic farming. So promote that along with education, or through education. And so some of the questions that we had, I worked with Dr. Kim Van Scoy in environmental studies, and also with Catherine Thompson, who was another environmental studies student. And so we were wanting to address how can we maximize integrated pest management and recruit more natural enemies. And then we wanted to increase the complexity by planting crops in polyculture with non-crops between rows. And so we were looking at this because there was discussion between using wood chips or filler crops in between rows in order to maximize production in garden settings, and using a mixture of monoculture and polyculture throughout the community. So people were already using these sort of methods, but unsure which ones were better. And so we wanted to help address these questions and bring this to general community, to citizens, and say, hey, if you want to maximize your crop production, then you can use these planting techniques. And so we wanted to look at which combination maximizes yields for our system. And so some of the literature suggests that increasing habitat complexity may increase natural enemy abundance. So just have more natural enemies present and residing near your situations or your systems. And then complexity provides refuges through that reduce intraguild predation, for instance, to stop predators from eating each other. And then this is given that herbivore density is not too low. And so we wanted to make sure that we address these issues. So we're hoping that herbivore density will be high enough in our systems to help make sure that we have enough of these natural enemies. And so some of the literature supporting this, so we have different types of habitat complexity with thatch present to create more or dead material, along with living material to create more complexity, with a lack of complexity or less complexity with thatch absent. And so we have predator density on the y-axis, and so we're looking at different kinds of predators present. And so even just looking at predator diversity, you can see that it's much higher or higher with with much higher looking at thatch complexity or having more individuals present. This created more diversity with thatch compared to less thatch. And you can see that with other predators as well, that having An increase in habitat complexity makes it so that there's more or of a refuge for different types of predators. And then as a result of this, you have a slight decrease with the amount of herbivores present in this area. So we're hoping that with increased habitat complexity, we'd also see this in our system. And just that overall architecturally more complex host plants have a greater diversity of herbivores. So applying this to our polyculture, having more a diversity of different kinds of host plants with increased diversity compared to monoculture, which is one kind of host plant. And so again, vegetation complexity provides a refuge at low prey densities. So even if you have a low prey system, you can have a refuge for those individuals to live in order to keep the predators from eating too many of each other, but it can still occur. So hopefully we're able to mitigate that in our system. So vegetation complexity provides a refuge at low prey densities for predators to reside until they can find higher prey densities. And so one of the hypotheses we had was that increasing IPM using higher plant architectural complexity, and this should increase natural enemy diversity and abundance. And so polyculture versus monoculture growing, planting schemes, mulch or green manure versus wood chips, green manure being sort of filler crops or non-crop plants in between plots. So our hypothesis being that greater habitat complexity through polyculture and green manure should increase natural enemy diversity. And so our other hypothesis was that, or our other aim was to look at how changes in planting and mulch influence crop yield. And so through natural enemy diversity, we should see a decrease in herbivores, which should therefore increase plant health and crop yield. And then so we also looked at polyculture versus monoculture, and then green manure versus wood chips. And so we thought that increased natural enemy diversity should increase crop yields via healthier plants. And so we were hoping that that would lead to higher plant or healthier plant metrics which I'll talk about in a second, that would lead to higher crop yields. And so here's a planting scheme where we had polyculture PC, monoculture MC, wood chips on one side, and then green manure on the other side, and with alternating planting schemes. And so 3 blocks per planting, MC or PC, plots surrounded by wood chips or green manure, and with our plots being about half a meter squared, and then 1 meter between blocks. And so there were either wood chips in between the plants, in between the plots, or there were this growing or noxious or harmless sort of weeds or non-crops in between these other plots. Monoculture was 4 Swiss chard plants. So Swiss chards typically grow in our area and by a lot of community gardens. So we want to focus on monoculture, which is 4 Swiss chard plants. And the polyculture was 1 Swiss chard, tomato, 1 bean, and 1 kale plant. And so a mixture of wide variety of complexities between these plots. And so for diversity, we converted Shannon's diversity index to true diversity. Natural enemy, herbivore, and omnivore diversity and abundance, but we focus more on natural enemy and herbivore diversity. And there's the equation that we use, converted it to true diversity. Sampled each week for 8 weeks. Arthropods identified as a family or species depending upon how common they were. There were a few taxa that were just present and not identified before they could disperse or take off, and so those were lumped into general categories. But the large majority of arthropods were either identified as a family or species if they're more common. And so that came down to me just saying everything that was there and writing down everything that I saw. So I walk up slowly to the plot and start looking for if anything was dispersing as soon as I got there. And then once I was in the plot, just writing down or identifying what I could on sort of site identification. And then if individuals were not able to be identified on site, then I would capture them and identify them in the lab using textbooks and also pest guides. And so for plant yield and health, we looked at the harvest, of course, but also we looked at approximate chlorophyll content, which is also a proxy for nitrogen content. We also looked at chlorophyll fluorescence. That would help us determine if soil systems were damaged and if they're able to maintain healthy levels despite any possible stress, or if they were healthy, we would see better or or a larger chlorophyll absorption of fluorescence. So if plants gave off more light as reflection, that means they were absorbing as much light for photosynthesis, and they're just reflecting it back. Or stressed plants can also use that light absorption for heat, which is another sign of stress. So we also looked at the weight of harvested foliage and fruit from our crops. And then we use the optosciences meter, OS 330P plus for quantum efficiency. That was for the fluorescence. And then the SPAD meter for chlorophyll, proxy for chlorophyll content. That looked at nitrogen content, just greenness of the plant. And then this was sampled each week, non-destructive sampling for 8 weeks. And we compared plots with all combinations. And so overall with the results, we actually identified 1,280 different arthropods from 46 different taxa. 13 of those groups were natural enemies and 13 were herbivores, and 12 omnivores, and a handful of detritivores, and a handful of sort of general taxa that didn't fit into any of those sort of groups. Stilt bugs, carpenter ants, wolf spiders, jumping spiders. We also saw lots and lots of sharpshooters, Cicadelidae. We saw harlequin bugs towards the end of the season, and we saw lots of aphids, primarily on the tomato plants. And we also saw garden cats. So we actually had a local cat that just lived at the garden in cauliflower that would show up every once in a while just to greet. So what we saw with natural enemy diversity, we saw more natural enemy diversity with monoculture. So surprisingly with monoculture. I will say though that we saw more healthier and larger monoculture with the Swiss chard. So again, monoculture was just Swiss chard plants. We saw larger amounts of Swiss chard plants within the monoculture, so that may have increased the architectural complexity in our system compared to polyculture, which slowly became dominated by the tomato plants. But we saw more natural enemy diversity in polyculture, so 77% more diversity within monoculture compared to polyculture. And so herbivore abundance was actually greater in polyculture and wood chip plots compared to other treatment combinations. And this was actually counter to a hypothesis which we saw, which we expected to have all this occurring in the polyculture. And also we saw natural enemy diversity lower in polyculture plots. So with higher natural enemy diversity in monoculture, we expect to see that where we saw lower natural enemy, lower herbivore diversity in polyculture, and also in monoculture. And we saw lower herbivore diversity. And then this is possibly due to, we also saw more herbivore diversity with green manure. So plants that were next to green manure actually supported more herbivore diversity. And that could have been just from the neighboring plants providing a refuge for our herbivores to live there and then come to our plots, totally possible as well. Whereas compared to wood chips, it was more of a landscape that they would travel, but it also supported or theorized to support, and this is through evidence as well that we've seen through other studies and also with other observations, that wood chips actually support more natural enemies as providing a better microclimate, and then retaining moisture much better during the summer compared to other plants. And so this is possibly due to natural enemy diversity being and abundance being higher in wood chip plots, which would actually lower the diversity within the wood chip plots. And then with the plant side, we saw that healthier plants were next to wood chips, and this is possible just with, we saw mostly with kale and Swiss chard that these individual plants were doing much better while next to wood chips. And then we also saw that kale had greater efficiency, so they were able to absorb light better and photosynthesize, sort of capture electrons and have higher photosystem efficiency when growing next to wood chips compared to green manure, which again might have been just a function of the plants nearby through competition or overshadowing, or just having a cooler environment for them to live next to compared to the green manure or other plants to compete with. So with plant yield as well, we saw higher yield with wood chips next to crops with green beans. And then we also saw more Swiss chard with wood chips next to them as well. So wood chips all around. This is also controlled for the amount of or Swiss chard when we get to the monoculture and polyculture. And then there's also more tomatoes. Of course, this is all looking at polyculture for beans and wood chips, but also when comparing and controlling for the number of Swiss chard plants between these two when looking at polyculture and monoculture. And then more tomatoes as well when looking at those grown to wood chips. And so we did this last year as well. So this was also in 2019. So we repeated the study with additional team members, Christina Waddle, which will also be in the ESA symposium or NSOC symposium in November, giving a talk on her research with Dr. Van Scoy, and then also Lindsay Cross, who also helped us collect data as well. And so the differences is that because of the results we saw from 2018, we had a reduction in the amount of space available for us because of other activities going on in the garden. So unfortunately, we had to decrease the number of plot sizes, but to help mitigate that, we tried to focus just on wood chips being the only factor, and then also polyculture and monoculture. And so we didn't have a mulch treatment of green manure versus wood chips. We just focused on just having wood chips between all the plots, trying to mitigate the reduced plot size. And then we saw the same hypothesis. And then for the plant side of it, again, just wood chips. But as a result of the changes, we had AmeriCorps out there. We also had other studies going on at the garden at the time as well. And so we actually had our plot number reduced to from 15 to 10. And then we had 7 weeks only instead of 8. So a few differences, which unfortunately made some impacts later. But we saw some differences compared to 2018. First of all, we saw a lot more diversity just overall compared to 2018. We had 2,031 arthropods identified this time, so higher abundance across 68 different taxa, so 27 different types of natural enemies or taxa, and then also 24 different kinds of herbivores, herbivore taxa, 6 omnivores, 4 detritivores, and then some individuals who were sort of in between or rare occurrences. We had a lot more cicadelid diversity this time around, and some brand new species that weren't in the first time around. So definitely an increase in diversity of these family members. And we also had no aphids. So very few aphids, a few appearances on tomato plants, but very few individuals of aphids this time around. Whereas last year, aphids actually had to be removed from part of the analyses because of their diversity. Not their diversity, but their abundance, the sheer number on some of the plants. And we also had more tobacco hornworms this time. And we also had earwigs and some other individuals like sosids that showed up this year compared to last year. We also had more arthropods. We had some millipedes, centipedes, and then also had a large turnout from other insects like ants. So more ant diversity as well. So some differences this year compared to last year. Which was interesting. And so what we saw a negative relationship though between natural enemy diversity and herbivore diversity across both plantings. So we weren't able to see this in 2018, but we saw this this year with just a correlation between the two. So a negative relationship. So we have increasing natural enemy diversity leads to also a decrease in herbivore diversity. Which we weren't able to show that last year. So we're able to show this year across both plantings. And so what we're able to see overall across these studies between both years, some of the highlights is that we found greater natural enemy diversity and abundance with monoculture and wood chips. And then we saw greater herbivore diversity and abundance with polyculture and green manure. And wood chips are actually an excellent matrix for natural enemy refuge. It seems to be the case. We've seen citizens and other community members have told us what they also use and what they've seen. And we've seen some studies looking at wood chips providing a much better refuge or through microclimates, especially as temperatures reach 40 degrees Celsius in the heat and sun during the summer in Arkansas. And so as a result of that as well, sometimes we'll see some integral predation as well as a picture I saw just spiders eating spiders. And so that could have been a result of a lack of herbivore diversity as it became too hot, especially in 2018, but also in 19. The prey population may have been too low in polyculture to support predators, leading to intraguild predation. And so that's why we may have seen a decrease. In 2019, we're able to show that relationship between natural enemy diversity and herbivore diversity compared to 2018. And this, however, we weren't able to see other factors. We may have to explore more of the data to see if we were able to find any more relationships because of the reduced study space. And both plantings had similar health metrics for 2018. This makes sense because if we have similar or reactions to or similar amounts of herbivore diversity and natural enemy diversity between both planting types, then we wouldn't see much between the metrics of the plants, then we wouldn't see much between the yield. But this is sort of just what we're piecing together in speculation at the time. So we'll have to explore more of the data to see if we can find more of those relationships. And for the plant side again, so we had greater yield with wood chips, beans, and Swiss. It was charted across the board, so wood chips are the way to go. And so we had more chlorophyll and efficiency in kale with wood chips, and then we also had tomato and green manure competition. So we may have seen tomato plants competing with green manure to influence that. And the wood chips may retain water and have advantageous microclimates, of course, and so that's more about speculation, but not just speculation, but also what we've seen with individuals and what we've seen with other studies. No relationships on the plant side, and maybe just because of the reduction in study size. And then we were able to see that new diversity, the increased diversity, which may have just changed relationships compared to 2018. All right, so I'm running out of time a little bit, so I'll wrap things up. But some things I'm doing in the classroom now that are looking at classroom undergraduate research experiences for students. So I'm working with Aaron, the Ecological Research and Education Network, and also NEON, which is a data repository, and also CUBE, which is a faculty mentor network that looks at doing these classroom experiences. So in my ecology evolution course, we're looking at how plants and their diversity and their woody and their woody sort of degree of woodiness, and also their diversity is influenced in human altered environments, so across urban gradients. And I'm also in my entomology course looking at backyard pollinators and how they are able to, how their diversity changes over an urban gradient, and how their functional ecology changes, and how their diversity changes over an urban gradient. And I like these because these are allowing us to look at, get students outside, looking at standardized plot design, randomization, and basic experimental design, and also identifying plants. And then the backyard studies looking at functional ecology and its role in pollination and how that can be affected by urban gradients. And so I know I went to that really fast, but I'm running out of time and going in my Q&A a little bit, so I want to make sure you all have questions. But some of these details, I like these studies in particular because it's allowing us to teach students how to identify as part of a entomology course, but also having them look at this urbanization gradient and how that has an impact on the degree of urban pollination and the functional diversity of different species. Okay, and with that, are there any questions?
Thank you so much, Warren. While people start to enter their questions in the chat, or you can raise your hand to unmute, we did get one. Oh, sorry, and put your questions in the Q&A. We got one in the chat from the first section. So, do you see memory? Are they more effective at fighting off an infection after a previous exposure in the pollinator work that you were doing?
Do I see what?
Memory?
Okay. So what we looked at, so we didn't look at or trace the members back to the colony to see if they were able to sort of remember and follow back to their hives. We just captured them right on the spot. And so when it comes to how the environment or how, if we can relate immunity to their ability to follow their way back to the hive, see if I think you're getting at the toxication of those individuals and how that may be a factor with immunity or their or their ingestion of toxic compounds, which may have affected their memory. We weren't able to see that. We didn't look at that in this study, but it would be possible theoretically with increasing ingestion of compounds, and if there's too much for. So one of the factors that will influence urbanization or urbanized pollinators is that degree of intoxication, and then being able to make their way back, how that neonicotinoids as pesticides, for instance, in urban areas has been shown to actually impact the memory of individuals as they make their way back. And so with those ideas, that theoretically would suggest that you'd have an impact in memory, but we didn't look at that in our study.
Thank you. And then we have one written here on the second part. Is there a way to reduce crop competition by the green manure so it doesn't reduce yield?
Yeah, so that was something that, of course, looking back was the issue, or one of the issues with 2018, that we had to actually periodically trim the green manure because that was just growing way too fast. And so we had to keep it trimmed. Almost every week we had to go out there and just trim it, or have the undergrads go out and just trim it in order to keep it low enough to reduce that competition. But one thing to do in the future, if we were to use this, so we do plan doing a third trial, and we would go back to I would like to look again at the green manure and the wood chips. But one way to mitigate that is to create or increase the distance between the plots and reduce that green manure. So that would use a section of green manure, but there's also some space that's just decreased or have a gradient of some sort, so that there's lower and shorter plants and fewer plants as you get to the plots that have our crops growing. And so I think that would decrease competition for nutrients, water, reduce, there were periods as well of overshading. And so that may also impact that. And so, yeah, definitely that was an issue that was also brought up before. So that would be something definitely to fix going into the third season. We were planning on doing a third season, but then COVID struck. So we got, roadblocked a little bit. But I may end up having to do it myself to maintain social distancing and next year. So.
And can you just remind us, in that study, you weren't using any sort of plastic mulching on the plants, correct? Or like row mulching, it was just bare soil that you were planting everything into?
Yes. So we did have debris from previous years or previous, the last growing season that was standardized across all the plots, but no plastic mulching. We just had wood chips on the outside, and then bare soil or leaf litter in the inside the plots.
Dr. Elizabeth Long shared a comment here. Thank you for sharing your work with us, Warren. I really enjoyed your seminar. Do you think crop variety might have played a role in the herbivore abundance diversity relationship? I wonder if some chards have slippery leaves, etc. Now I want to do a mini experiment in my raised beds at home because chard is delicious.
Yes, we did enjoy the harvesting part quite a bit of that study. Looking at your question again, just real quick. Yeah. So some part of the data, especially in 2019, we did see a lot of individuals who are sharpshooters and other psychedelics who were all about the Swiss chard. And yeah, we probably did see a completely different system or different response if we just changed that variety of crops, even different species or different tomato varieties. Because in polyculture, the tomato, especially in 2018, it would grow quite a bit. And so you have plots that started to become monopolized by the tomato as just its natural growing pattern. And so even changing the tomato crop variety would have altered that as well. And then also the Swiss chard ended up there were other herbivores on there, but primarily it was sharpshooters and other psychedelics. So that was the main issue. So it's possible that if we had different kinds of chard or just replace that monoculture or Swiss chard with something else, we may have had a different diversity, of course, as some interactions are going to be crop and species specific. But I definitely think crop variety played a role. And we use these particular combinations because those are common crops in our community gardens and what people grow in our area. And so we wanted to directly relate our results to what they do and have it applicable for the everyday gardener.
Thank you. Maybe one last question can come in if you want. Otherwise, I know graduate students, you will have an opportunity to interact with Warren and ask him some questions during the lunch hour. So I just want to say thank you again, Warren, and I look forward to talking to you later. And thank you everyone for attending.
Thank you. I had a good time.